Alright, so today's mini lecture is on Gregor Mendel and the very important contribution he made to our understanding of biology and genetics. Okay. Here are the key objectives. Again, this is a review topic for ninth graders because the introduction to genetics has been covered already in middle school. So really we're just emphasizing the use of the vocabulary and in being able to do these Punnett square problems, which we will spend a lot of time on in class. Alright, so Gregor Mendel, he was an Australian monk and basically he worked out in the garden, and to keep himself busy he started playing around with the different traits of garden peas. Okay, he is known as the father of genetics, which is the scientific study of heredity. And I would rank Gregor Mendel probably the third most important person in all of biology. Okay, we definitely learned a lot from him, and even though we found out later that there were some things that he said that were not exactly true, he gave us a starting point, and certainly his laws are still upheld today. So pea plants normally reproduce asexually. In other words, the male parts fertilize the female parts. This is known as true breeding. Therefore, they would produce the identical offspring to their parent to the parent plant. So in other words, there would be no genetic variation whatsoever. If you had a tall plant, you would produce more tall plants. If you had a short plant, you would always produce more short plants. And even more specifically on a genetic level, they would have the exact same DNA. Okay. However, and this is what Mendel did, we can make plants reproduce sexually. Okay, and we do this by crossbreeding them. Physically, what we do is we collect the pollen, which is essentially plant sperm. We collect the pollen with like a paintbrush or something, and we brush it on the female reproductive organs of another plant. So if the pea plants are cross-pollinated through our crossbreeding, then what type of offspring would they produce? Because now we know there's a chance for that recombination to occur since those gametes are going to be joining together through fertilization. So a trait is what we use to describe the physical expression of something that we see on a genetic level. So for example, I have a trait of having blue eyes. So my eye color is the trait, okay? You also have eyes, you have color to your eyes, but your eyes may be green or brown. So if we stick with some plants, examples, seed color, seed shape, the seed coat, and also plant height. One thing that Mendel did very well, as every scientist should, is that he kept documentation of all of his work. So he set up his procedure and he was very organized. When you do this in an organized fashion, you're going to divide up your generations, and what he did is he named them. So the first plants that he would cause crossing to occur between, he would call the P generation, or P for parental. Then any offspring that they made, those were called the F1 generation, first filial generation, and filius is the I think it's Latin. Filius is the Latin word for son. So that's where he got the idea. So P generation is at the top. The babies that they make are going to be called the F1 generation. Okay? If we take two plants that are not genetically identical to each other and we crossbreed them, the offspring that result from that crossbreeding are going to be called hybrids. And that makes sense if you think about, where the word hybrid pops into your head. Think about the popular car. Okay, it is a hybrid between running on gasoline and electric. So it contains traits of both a gasoline-powered car and an electric-powered car. So Mendel did this experiment. He crossed tall with short. All offspring were tall, and we're talking like 300-400 offspring. He crossed green with yellow, and all of the offspring were green. He crossed round and wrinkled, and all of the offspring were round. So what he concluded was that all of the offspring took on the trait of one of the two parents. Okay. How did he feel about this? He was kind of shocked, and these are actually all of the traits that he looked at. Seed shape, seed color, seed coat color, pod shape, pod color, flower position, and the height of the stem. Okay, so first thing that he concluded after he got over his confusion, I should say, is that the traits in the offspring are being controlled by something that gets passed on to them from the parents. We know that this is on their DNA, and we've talked about this when we looked at meiosis and how those gametes are formed from each parent and then how we fuse them together through sexual reproduction. But he didn't have access to DNA. They didn't even know what DNA was at that time. So he just called them inheritance factors. We now know that those factors are called genes. Okay? There are different forms of genes that you can have, and the word we use for that is allele. So, for example, if we're looking at the gene, which is plant height, okay, we can have an allele which is for tallness, or we can have an allele that is for shortness. Okay. The other thing that he concluded is this idea known as the principle of dominance, that some of the alleles have more power than other alleles, and so he used the words dominant and recessive to define their difference. If an allele is dominant, we're going to represent it with a capital letter when we denote an organism's gene pairing, what they have on each chromosome. So the dominant allele will always be expressed. That means if they have a dominant allele, they will show the dominant trait. They only need one in order for this to be the case. Okay? However, if they are recessive, the only way you will see the trait from a recessive allele is if there is no dominant allele present. And we're going to use a lowercase letter here to represent that. So we know for every gene, we have two copies of it that come to us, right? One comes from the egg gamete, one comes from the sperm gamete. And together, that's what makes our homologous pairs, right? Chromosome 1 from dad, chromosome 1 from mom. And what that's going to mean is, for example, with these plants, they inherited a dominant allele from, the mother, okay? And let's say they inherited a recessive allele from the father. So when we look at the gene pair together of what they have one dominant allele and one recessive allele. So what is their phenotype going to look like? Well, since they have a dominant allele present, then that means that the trait for the dominance will be expressed. So if that is, tall, then the plant will come out tall. Okay? When we look at these gene pairs, right, because you have two alleles for each gene, we can have different combinations. They can be homozygous, which means you inherited two of the same alleles from your mom and your dad. They could both have been dominant, which would give you big T, or they both were recessive, which would give you little t. The other option is that you were heterozygous, which means you inherited one dominant allele from a parent and one recessive allele from the parent. So when we describe what your traits are on a genetic level, meaning looking at the alleles, the letters that we see here, that is known as the genotype. So if I said, the genotype for having a recessive condition is little t. Whereas the phenotype, that would be a word, that would be the physical trait and how it's expressed, what we see it as. In this case, little t, the phenotype would be small. Okay, so here we're putting it all together. If you have a dominant homozygous genotype, okay, then your phenotype will be tall. and you will have a genotype big T, big T. If your genotype is big T, little t, your phenotype will still be tall because remember, the presence of that dominant allele, the big T, it covers up the expression of the little t, so it can't show itself. It's sort of like it overpowers it, okay? And you would be considered heterozygous, and therefore you are tall. And lastly, if your genotype is little t, two recessive alleles together, they're going to be able to express the recessive phenotype, which in this case is short, and we would describe that as homozygous recessive. Okay? So Mendel's last question, he knew that there were recessive alleles in the parent generation because he crossed one tall parent and one short parent. So he wanted to know what happened to the recessive alleles from that short parent since all of the offspring were tall. He wanted to know, did they disappear or are they still there? So what he did is he mated together two of the F1 plants from that cross. Okay, so we started with the P generation. We had our pure tall and our pure short, and we created all tall offspring. He took two of those offspring and mated them together to produce the F2 generation, and these were the results we found. Okay, for every trait that he was looking at, if the F1 generation showed all of one trait expression, in the F2 generation, there was now a split. 75% showed that expression and 25% showed the other form. So when we get all the way down to the bottom, if we make this generic, it says their trait X, okay, whatever trait you're looking at, if you have a dominant crossed with a recessive and they're purebred, all of your F1 offspring will be dominant. Okay, but then when you take that F1 offspring and mate it with another F1 offspring, your F2 generation will show 3/4 dominant and 1/4 recessive. Again, this does not hold true for all of the traits that we have. It only holds true for some of them, the ones that Mendel was able to prove. But the pea plants turned out to be a really good example plant. Okay, so we take a tall plant and we mate it with a short plant, and all of the F1 generations are tall. Then we cross them together, and if we look at our ratio, we would have 3 tall plants to 1 short plant, or 75% would be tall to 25% being short. So how can he explain this? It's not that the recessive alleles disappeared, but at some point when we were forming our gametes, that allele for tallness was able to separate itself from the allele for shortness. Okay, we've already talked about segregation when we talked about meiosis in the previous section, but let's take a look at it again. Okay, so in the F1 generation, the parents had big T little t and big T little t, and we know that, here, let me just add a little fact here. Okay, we know that because our parents, we had one true breeding tall parent and we had one true breeding short parent. So when they mated together to produce the F1, okay, if we make a combination, the only possible combinations we have here are that the tall parent, ooh, that got a little long, sorry, that the tall parent donated a dominant allele and the short parent gave a recessive allele. So all of our offspring had to be heterozygous. So therefore we take one heterozygous individual and mate it to the other, and then when they form their gametes for that mating to produce the F2, well, you could give either a big T or a little t in your gametes. Okay, these would be the gametes formed. Oops. E, T, E, S. Okay, and these are the gametes for this individual. All right. So when we look at the chances of these gametes coming together, these are all the possibilities, these 4 right here. Okay, so if we look at all 4 of them, of the 4, 1, 2, 3 of them would give us a phenotype of tallness, right? Double homozygous, dominant, and then both of these are heterozygous. So we would still see that 3-to-1 ratio. Okay. So again, like I just said, looking at the plant height, the F1 plant had the genotype of big T, little t. When that plant formed gametes, you can only choose one. So we divided into 4 cells. 2 of the cells gave a big T allele, and 2 of the cells gave the little t allele. And that's how we were able to discern what type of gametes would be formed, and then therefore that's how we got that chance of having that recessive offspring come back. So let's take a look back at that diagram again. This would be a good diagram to make a sketch of. I would say let's put it right here on the left-hand side in your notes. Okay, put it right here. Make a sketch of this so that you have an understanding. Okay, and again, this is a review of what we had talked about in meiosis. We know when those tetrads line up on the metaphase plate during meiosis, they will eventually be separated in anaphase 1. And so that separation now allows them to be expressed individually. And like the recessive allele, if they happen to be combined with another recessive allele, then they will have the chance to show. So it's not that the recessive allele disappeared, it was just being masked by the fact that the dominant allele was always present. Okay, that's the end of the notes. If you have any questions, please email me or see some of the links and resources on the website.